Hello friends, welcome to my channel Easy Life Science. Here I am talking about some important questions, answers with a detailed explanation. The questions which are asked for a set life science exam. So subscribe my channel Easy Life Science, like the videos and share it to your friends. Don't forget to press the bell icon so you will get the notification of my newly uploaded video. So let's start. The question first. DPN first is used to digest the PCR products during site directed mutagenesis because the DPN first digests only a methylated DNA. The option C is the answer of this question. DPN first is a type 2 restriction endonuclease. Source for this enzyme is a bacterium, that is a Diplococcus pneumoniae. DPN first is specific for methylated and hemimethylated DNA. Since DNA isolated from E. coli strain is damn methylated, it is susceptible to DPN first digestion. Hence, DPN first frequently used after a PCR reaction to digest the methylated parental DNA template and select for a newly synthesized DNA containing mutations. So it is used in a PCR for a site directed mutagenesis as it digests only a methylated DNA. So option C is the answer for this question. Which one of the following is an incorrect biomolecular modification pair? The biomolecules get modified. The RNA after a transcription process, number of adenine units are joined to a 3' ends of the RNA. So this is known as a polyadenylation. This is a modification of RNA. The proteins are modified N-linked glycosylation or O-linked glycosylation, whereas the lipids are modified by a palmitylation, myristoylation or a perenylation. So option A, option C and option D are a correct pair, whereas option B is incorrect. The DNA get modified through a process of a methylation, whereas the proteins does not modify a methylation process. So option B is the answer for this question. By mistake it is ticked as option A, so recorrect it as option B. Next question, a mixture of following purified proteins IgG, IgM and Fab fragments of immunoglobulins is separated using a gel filtration chromatography. The order of elution of this protein first to last is. Here in this question, the question is asked on a gel filtration chromatography. The gel filtration chromatography is a technique which separates the proteins solely on the basis of a molecular size. The separation is achieved using a porous matrix. To this porous matrix, the molecules can access with a different degree. Small molecules have a greater access and larger molecules are excluded from the matrix. In this gel filtration chromatography, the molecule which having a high molecular weight get eluted first. So as the IgM has a high molecular weight, it get eluted first. Then the IgG has a low molecular weight than a IgM, and then a fragment for a antigen binding. So sequence is the IgM, IgG and a Fab. So here in this image, the 5 antibodies are there. The molecular weight for the IgM antibody is 9 lakh Daltons. The molecular weight for IgG is 1,50,000. For IgA 3,85,000. For IgE 2 lakh and for IgD 1 lakh 80. So the question is asked for first to last. The high molecular weight Ig immunoglobulin is the IgM, so it will get eluted first. IgM, then the IgG, and then the Fab fragment. In a same image, some differentiating characters are mentioned here. The heavy chain of IgM is called as a mu. Heavy chain of IgG is a gamma. Heavy chain of a secretory IgA antibody is a alpha. Heavy chain for a IgE is a epsilon, and a heavy chain of a IgD antibody is a delta. Number of antigen binding site for a IgM 10, for IgG 2, for IgA 4, for IgE 2, and for IgD 2. The IgM, it is a main antibody of a primary response. It is common for a complement fixation. Then the IgG, the main antibody in a blood, it neutralizes a toxin and responsible for opsonization. Then IgA, this is called as a secretory antibody, secreted into a mucus, tears, saliva, and a colostrum. Then IgE, it is also called as an antibody of allergic response. And a IgD, it is known for a B cell receptors. So for this question, answer is a IgM, IgG, and a Fab fragment. This is a sequence of elution after a analysis through a gel filtration chromatography. Then the next question, ascending order of a half-life for radioactive isotopes, iodine, hydrogen, carbon, and phosphorus. These are the 4 radioactive isotopes, and you have to arrange them in ascending order of their half-life. The half-life of phosphorus, it is a 15 days. Phosphorus 32, the half-life of this radioactive isotope is a 15 days. Then the half-life of iodine 125 is a 125 days. Half-life for a carbon is a 12.4 years, and half-life of carbon 40 is a 5730 years. So on ascending order, the phosphorus, then iodine, then hydrogen, and then carbon. Option D is the answer for this question. Then the next question, Rubisco catalyzes a conversion of ribulose 1,5-bisphosphate 2. 4 options are there. The Rubisco, it is a enzyme which is necessary for photosynthesis. It is a largest protein or a enzyme on a universe, which is abundant in a universe. The ribulose 1,5-bisphosphate carboxylase oxygenase, it is a long form of Rubisco. Ribulose 1,5-bisphosphate carboxylase oxygenase, and this Rubisco enzyme fix a carbon dioxide to a energy rich molecule, that is a glucose, during a photosynthesis. Substrate for a Rubisco are ribulose 1,5-bisphosphate and a CO2 in a photosynthesis, or ribulose 1,5-bisphosphate and a O2 in a photorespiration. In a photosynthesis, Rubisco catalyzes a first step of a carbon fixation in a C3 pathway or a Calvin cycle, that is carboxylation of ribulose 1,5-bisphosphate, and because of the carboxylation of ribulose 1,5-bisphosphate, it results in the formation of 2 molecules of 3-phosphoglyceric acid. This 3-phosphoglyceric acid is a 3 carbon containing molecule. So in a photosynthesis, because of a carboxylation reaction by a Rubisco, 2 moles of a 3-phosphoglyceric acid are produced. In a photorespiration, Rubisco has an affinity for oxygen and it oxygenates ribulose 1,5-bisphosphate in presence of oxygen. Photorespiration utilizes a ATP and hence it leads to wasting of some energy produced in a photosynthesis. When Rubisco binds to a O2, it converts Rubisco to it converts a ribulose 1,5-bisphosphate to one molecule of phosphoglycerate and one molecule of phosphoglycolate. The phosphoglycolate is a 2 carbon containing compound. Photorespiration occurs in a light. It consumes oxygen and produces a CO2 just like a mitochondrial respiration. So Rubisco has two activity. One is a carboxylation and another one is a oxidation. In a photosynthesis, it forms 2 moles of a stable compound, that is a 3-phosphoglycerate, and in a photorespiration it forms a 1 molecule of 3-phosphoglycerate and a 1 molecule of phosphoglycolate. So option P and Q are a correct option for this question. Rubisco catalyzes conversion of ribulose 1,5-bisphosphate to 2 moles of stable 3-phosphoglycerate and another 1 molecule of 3-phosphoglycerate and 1 molecule of phosphoglycolate. Then moving to the next question, here you have to match the correct pairs from group A to group B. In a group A, ubiquitin phototropins. This is a phototropin, not a phytotropin. This is a phototropin. Extensins and delaproteins. In group second, cell wall loosening enzymes. Second, repressor in a gibberellin signaling, mediator of protein degradation, and fourth, non-competitive inhibitor of polar transport of oxygen. The first, that is a ubiquitin. We know that the ubiquitin is a small protein made up of 76 amino acids and it is a regulatory protein. It is present in all eukaryotic cell, directing the movement of important protein in a cells. Ubiquitination affects a cellular process by regulating the degradation of a protein via a proteasome and a lysosome. Ubiquitin is a small protein. It links to a targeted protein through a lysine residue, and that after ubiquitination this protein get degraded via a proteasome. So for ubiquitin, the from group second, third pair is matching the mediator of a protein degradation. Then Q, phototropins. The phototropins or a phototropism means movement in a response to a light. We know that the auxins are a plant hormones responsible for a cell elongation in a phototropism. When light is coming from one side of the plant, it diffuses toward the shady side of the shoot, which stimulates the cells to grow longer, resulting in a bending of a shoot toward the light. Thus, oxygen promotes a phototropism. So, for a group A, to a phototropins, the fourth from group second is matching non-competitive inhibitor of a polar transport of oxygen. Then R, extensins. The extensins are a plant cell wall hydroxyproline rich glycoproteins known to be involved in a cell wall reinforcement in a higher plants and in a defense against a pathogen attacks. So for extensin from group second, the first pair is matching cell wall loosening enzyme. Then yes, that is a delaprotein. The delaproteins are a repressor in a gibberellin signaling. The gibberellin is a plant growth hormone, controls major aspects of a plant growth such as germination, elongation, flower development. The DELA proteins are involved in a gibberellin signaling and they are negative regulation of a gibberellin signaling. So means they are a repressor of a gibberellin signaling. Next question here, some plant species are there, their family and plant products. P. Cannabis sativa, it is from a family Cannabiaceae. And their leaves are useful for production of marijuana. P24. Then a Q, Corcorus olitorius. The family of this Corcorus olitorius is a family Tiliaceae, and the fibers are produced from stems of this plant. Then R. Osmium sanctum. This plant is from family Lamiaceae, and from leaves of this plant there is a production of eugenol. Then plant species, yes, Ricinus communis. This plant from a family Euphorbiaceae, and their seeds are useful for a production of oil. So option D is the answer for this question. Then the next question, hybrids may arise through the hybrids are produced by combining a cytoplasm from nucleated cells with non-nucleated cell or a cytoplast. The nucleated cells can be unaltered or could have undergone a depletion of endogenous mitochondrial DNA before a cytoplasmic mixing. The hybrids are important for the transfer of cytoplasmic male sterility towards the resistance from antibiotic herbicides. So, the hybrids may arise through elimination of one of the nuclei from a heterokaryon formed from two protoplasts of a different origin. Option C is the answer for this question. Here is the explanation of the Rubisco. When a Rubisco has a carboxylase activity, it forms 2 moles of a 3-phosphoglycerate in a Calvin pathway, but in a photorespiration, when it binds to O2, it has a oxygen activity and forms 1 molecule of 2-phosphoglycolate and 1 molecule of 3-phosphoglycerate, and then the glycerate and a glycolate further get converted into a number of amino acids, whereas from Calvin cycle this 3-phosphoglycerate get converted into a glucose through a reversal of a IMP pathway. The plant cells are considered totipotent. The phenomenon of mature cell reverting to the meristematic state and forming undifferentiated callus culture is called. Plant cells are considered as totipotent. The phenomenon of mature cell reverting to a meristematic state and forming undifferentiated callus tissue is called. This is called as a de-differentiation. We know that the differentiation, starting from a differentiation, it is a process in which cells and a tissue gains several characteristics to perform a specific function. Once a cell is differentiated, it becomes committed to particular fate and start their function. But in de-differentiation, the de-differentiation is a process by which matured or specialized cells lose their differentiated characters and rejuvenate, or It is a process in which tissues that have undergone a cell differentiation can be made to reverse the process so as to become primordial cells again. And the term totipotency, it is the ability of a single cell to divide and produce all the differentiated cell in an organism. The option C, that is organogenesis. It is a process of development of organs like shoots, roots, flowers, either directly from a explant or from a callus culture. The embryo is not considered as a organ because in an embryo there is an absence of vascular system. So answer for this question is a D, differentiation. In a D differentiation, the differentiated cell get converted into a meristematic state, or it is converted into a primordial state. So answer of this question is a D, differentiation. Here in this image, these phenomena are explained. Suppose one cell is there and it get its fate and it carry out its function by a process, that is a differentiation, and when these differentiated cells are reverted to a meristematic state, it is called as a de-differentiation. This trans de-differentiation The next question, glyphosate is a broad spectrum herbicide. Upon application to leaves, it is translocated to a meristematic area and underground rhizome by the glyphosate, that is a Roundup, is a broad spectrum herbicide. It is absorbed through a foliage and through roots and transported to a growing plant. It inhibits a plant enzyme involved in a synthesis of three aromatic amino acids, that is tyrosine, tryptophan and phenylalanine. So it inhibits a growth of herbs and it gets translocated to a underground rhizomes or a root through a phloem system. So option A is the answer for this question. Here different in between de-differentiation and re-differentiation. De-differentiation, it is a reversal of differentiation, whereas re-differentiation is a reversal of de-differentiation. Through de-differentiation, the cell that matured and lost its capacity to divide regains a capacity to divide. In a re-differentiation, the cell that regained the capacity to divide loses its capacity to divide. Example of de-differentiation is that matured cell become meristematic cell. In a de-differentiation, matured cell that become meristematic cell through a de-differentiation again become a matured and specialized. Formation of cork cambium from fully differentiated parenchymal cell, it is an example of de-differentiation. Secondary xylem and a secondary phloem are the example of a re-differentiation. Then the next question, which of the following ecosystem is represented by an inverted pyramid of a number? In an inverted pyramid, the broad is very small, whereas the apex is large. So it is called as inverted. An inverted pyramid is formed when the number of individuals or a biomass is minimum and increases at each trophic level. The pyramid of a biomass in a pond ecosystem is an example of inverted pyramid because the biomass of phytoplanktons may smaller than that of the zooplanktons. The forest ecosystem, The pyramid of a number is inverted because the number or a biomass at the producer level is a minimum and increases at each trophic level. So, the ecosystem, that is a forest ecosystem or a parasitic forest ecosystem, is an example of inverted pyramid. Then the next question, rhyophytes are the deposits of the many plants accumulate a calcium oxalate crystals in response to a surplus calcium. So the rhyophytes are the deposits of a calcium oxalate. Answer of this question is option A. The next question, which of the following families shows bicollateral vascular bundles in a transverse section of the stem? The bicollateral vascular bundles are found in a family Cucurbitaceae, Solanaceae, Apocynaceae. The central column of the axis of the root and stem is called as a stele. This stele is made up of a number of vascular bundles which constitute vascular tissue systems like xylem, phloem and cambium. The arrangement of a vascular bundle are of three types: radial, conjoint and bicollateral. In a radial arrangement of a vascular bundle, xylem, phloem are arranged in an alternate manner on a different radia. All the roots of the plants contain radial vascular bundles. Then a conjoint, when a xylem and phloem are present on some same radius, it is called as a conjoint vascular bundle. Then a bicollateral, in this type two patches of a phloem, one each of the side of a xylem are present. In such vascular bundles, there are two strips of a cambium on each side of a xylem. Such bicollateral vascular bundle are present in plants of a family Cucurbitaceae, Solanaceae and Apocynaceae. So option C is the answer of this question. Then the next question, arbuscules are highly branched structures formed in or formed by. The arbuscules are formed by endomycorrhiza. They have a number of significance for the plant. Arbuscules are a site of nutrient exchange between a plant and the fungi. The nutrients like phosphorus, carbon, sulfur, nitrogen and a micronutrient are transferred from plant and a fungi. The arbuscules give resistance to a plant towards the number of abiotic trace. They protect a plant from metal, trace and unfavorable temperature. They help absorbing more nutrient from soil during a nutrient deficiency or at time of starvation. The arbuscules help in maintaining a salt concentration. Arbuscules protect the plant during a drought condition. These are the abiotic trace. Whereas the arbuscule gives resistance from a biotic trace, they provides resistance to the pest and insect by releasing volatile compounds. The hyphae of arbuscule decreases a nutrient content of weeds and hence working as a weed killer. Due to colonization, no site left for a pathogens to act. Colonization by arbuscule mycorrhizal fungi, it is useful for a plant. So, the endomycorrhiza forms a arbuscule on a plant. The next question, which one of the following combination of a polysaccharide is present in a plant cell wall? The plant cell wall is made up of cellulose, hemicellulose and a pectin. Option C is the answer of this question. Then next question, which of the following statement is incorrect? Option A, nitrogen fixation is an anaerobic process, and this is incorrect because the enzyme nitrogenase works at absence of O2. This nitrogen fixation is an anaerobic process. In option A, it is said that nitrogen fixation is an aerobic process, so it is an incorrect option. Other option, the dinitrogenase catalyzes reduction of nitrogen to ammonia. It is correct. Root nodules are formed in a glycine mac, that is in a soybean. It is also correct. The nitrogen fixation is an anaerobic process. Yes, it is also correct because the enzyme nitrogenase works better at absence of O2. Here in a question, it is asked that incorrect statement, that is option A. It is an incorrect option or an incorrect statement. Then next, which one of the following statement is incorrect with respect to a cyclic photophosphorylation? The cyclic photophosphorylation two types of photophosphorylation are there, cyclic one and a non-cyclic one. The non-cyclic photophosphorylation is termed as a Z scheme. In a cyclic photophosphorylation, the electron from a chlorophyll pigment or a photon get reverted back to a chlorophyll. So it is termed as a cyclic. But in a non-cyclic, the electron does not turn back to a chlorophyll. It is accepted by a NADP. Number of carriers are there in this photophosphorylation. In a cyclic photophosphorylation, the excited electron or a high energy electron from a chlorophyll is accepted by a peridotropin to a plastoquinone to a cytochrome, and cytochrome to a plastocyanin, and plastocyanin to the once again reverted back to a chlorophyll. So this is a cyclic process. In a non-cyclic process, photosystem second or a chlorophyll of a photosystem second, that is a chlorophyll 680, it generates a high energy electron. It is accepted by a plastoquinone to a cytochrome to a plastocyanin to a chlorophyll in a photosystem first, that is a chlorophyll 700. Then excited electron accepted by a ferredoxin, then ferredoxin to a NADP, and NADP to a NADPH. Here the electron does not goes backward to a chlorophyll, so it is called as a non-cyclic. Here are some differences in between cyclic photophosphorylation and non-cyclic photophosphorylation. Cyclic photophosphorylation, it is performed by a photosystem first. There is a working of only one photosystem, that is a photosystem first. Whereas in a non-cyclic photophosphorylation, it is performed by a collaboration of both photosystem, that is photosystem second and first. In a cyclic photophosphorylation, external source of electron is not required. In a non-cyclic photophosphorylation, it requires an external electron donor. In a cyclic photophosphorylation, there is no any photolysis of water, therefore oxygen is not evolved. In a non-cyclic photophosphorylation, it is connected with a photolysis of water and carry out a liberation of oxygen. The cyclic photophosphorylation synthesizes ATP only, but in non-cyclic photophosphorylation, in addition to ATP, there is a production of a reducing power in form of NADPH. Cyclic photophosphorylation operates under a low light intensity at an anaerobic condition or when CO2 availability is poor. The non-cyclic photophosphorylation takes place under an optimum light at aerobic condition and in presence of a high amount of carbon dioxide. The cyclic photophosphorylation does not take part in a photosynthesis except in a certain bacteria, for example purple sulfur bacteria and a non-purple sulfur bacteria. The non-cyclic photophosphorylation system is connected with a CO2 fixation. The cyclic photophosphorylation mostly occurs in a stromal or a intergranular thylakoids. the non-cyclic photophosphorylation occurs in a granule thylakoids. This ATP synthesis in a cyclic photophosphorylation is not affected by a DCMU. The DCMU stands for a dichlorophenyl dimethylurea. But the non-cyclic photophosphorylation ATP synthesis is inhibited by a DCMU. Once again, here in this image, you can look for the differences in between cyclic photophosphorylation and non-cyclic photophosphorylation. In cyclic photophosphorylation, only one photosystem involved, that is a photosystem first. In a non-cyclic photophosphorylation, PS1 and PS2 are involved. The reaction center is P700 in a cyclic photophosphorylation. The reaction center is a P680 in a non-cyclic. Electron releases are cycled back in a cyclic one, but in non-cyclic photophosphorylation, electron released are not cycled back. Photolysis of water does not take place in a cyclic photophosphorylation. Photolysis of water takes place in a non-cyclic photophosphorylation, so O2 is released. Only ATP is synthesized in a cyclic photophosphorylation. In a non-cyclic photophosphorylation, ATP, NADPH and hydrogen are synthesized. Photophosphorylation in a cyclic photophosphorylation takes place at two steps, whereas in a non-cyclic phosphorylation takes place at only one place. Cyclic photophosphorylation does not require an external electron donor. Non-cyclic photophosphorylation requires an external electron donor like H2O or H2S. Sensitive to a dichlorodimethylurea, whereas a non-cyclic photophosphorylation is sensitive to a DCMU. So here in this question, the question is asked the incorrect statement. ATP is generated without a commitment formation of NADPH. It is a correct statement. In a cyclic photophosphorylation, only ATP is produced, no NADP is produced. Statement B. Electron flows from a photosystem first to a cytochrome BF complex. It is a correct statement. Photosystem second does not participate in a cyclic photophosphorylation. It is also a correct statement. In a cyclic photophosphorylation, there is no any involvement of a photosystem second. Option D or statement D. Cyclic photophosphorylation occurs when NADP to NADPH ratio is high. There is no question of a NADP or no any matter for a NADP presence or absence. So option D is the incorrect statement for this question. Then next question, the light harvesting pigment not used by a blue-green algae or a cyanobacteria for photosynthesis is. The cyanobacteria does not make a use of a rhodopsin for a photosynthesis. Cyanobacteria make a use of phycobilins, phycoerythrobilins and phycocyanobillins as a photosynthetic pigments. So option A is the answer for this question. The next question, slow intravenous infusion of ethanol is a therapy to treat a methanol poisoning. The underlying chemical reaction is an example of. It is an example of a competitive inhibition. Ethanol or fomepizole. This serves as a alcohol dehydrogenase inhibitor. They stop a conversion of methanol to its toxic metabolites, that is a formate. When alcohol dehydrogenase is inhibited, then the methanol is also cleared. In a ethanol pathway, acetaldehyde is broken down into a acetic acid or a vinegar. This is a non-toxic product is then excreted safely. However, in case of methanol, formaldehyde is converted into a formic acid and formic acid deprives O2 of a cell and causes them to die. So methanol is a danger for a cell, and when ethanol is used as a therapy to treat a methanol poisoning, ethanol acts as a competitive inhibitor of alcohol dehydrogenase, and hence the ethanol get converted into a acetic acid rather than a formic acid. So it is a competitive inhibition of a enzyme alcohol dehydrogenase. So option A is the answer for this question. The next question, nitric oxide synthesis is responsible for a generation of a nitric oxide, an important signaling molecule. The substrate for this enzyme is nitric oxide synthase. This is a group of enzyme, or it is a family of enzyme, and this enzyme catalyze a reaction for a production of a nitric acid from a arginine amino acid. The nitric acid, it is a neurotransmitter and it is important for cellular signaling. It helps to modulate vascular tone, insulin secretion, peristalsis, and neural development. So substrate for this enzyme nitric oxide synthase is arginine amino acid. Then next question, allergies are due to hyperimmunoresponse. Drugs given to counter allergy targets the allergy are due to a high response of antibody, that is IgE, and this antibody produces the histamine, and to cure the hypersensitivity the drugs target for a histamine. For example, the drug like cetirizine are also called as antihistamine drugs. So option for this question is a histamine is a target for drugs at time of allergies. Then next question, catalase is found exclusive in. The catalase is an enzyme. It breaks down toxic hydrogen peroxide which is formed at time of respiration. In aerobic bacteria, there is a production of hydrogen peroxide, but this hydrogen peroxide through activity of a catalase get break down into hydrogen and oxygen. So toxicity of hydrogen peroxide is nullified by a catalase by a aerobic organism, but for an anaerobic organism, when these organisms are kept in a oxygen or aerobic condition, the oxygen for anaerobic organism become toxic as there is a production of hydrogen peroxide and lacking a enzyme catalase to break down this hydrogen peroxide. This organism or anaerobic organism does not grow in presence of oxygen, rather than they get killed. Oxygen becomes a toxic for them because of absence of a catalase. This catalase is formed in peroxisomes of the organisms. Option C is the answer of this question. The next question, RAG recombinase is responsible for formation of specific immunoreceptor. This process occurs in a T cell and a B cell. The RAG recombinase, RAG first and RAG2 recombinase catalyzes all the VDJ recombination events. The RAG proteins initiate a recombination of the variable V, diversity D, and joining J genes at T cell receptor, that is a TCR, and immunoglobulin loci of VDJ recombination process, allowing for a generation of T and B cell with a broad antigen recognition specificity. So option A is the answer of this question. The next question, example of substrate level phosphorylation. In a glycolysis, there are the two example of ATP generation through a substrate level phosphorylation. One is a conversion of 1,3-diphosphoglycerate to a 3-phosphoglycerate, and another one is a formation of a pyruvic acid from a phosphoenol pyruvate. These are the two examples in a glycolysis where there is the production of a ATP from a substrate level phosphorylation. Also in a tricarboxylic acid cycle or a TC cycle, succinyl coenzyme A get converted into a succinic acid, and there is the production of a ATP molecule or a JTP molecule. This is also an example of a substrate level phosphorylation. But in this question is asked in a glycolysis. So conversion of 1,3-diphosphoglycerate to a 3-phosphoglycerate is an example of substrate level phosphorylation. Here in this image, the one inorganic phosphate from a substrate is accepted by a ADP, and ADP get converted into a ATP. This is called as a substrate level phosphorylation. In this image, the phosphoenol pyruvate get converted into a pyruvic acid, and one inorganic phosphate is accepted by a ADP and leads to formation of a ATP. This formation of ATP is a substrate level phosphorylation. Substrate level phosphorylation is a metabolic reaction that results in a production of a ATP or a GTP by a transfer of a phosphate group from a substrate directly to a ADP or a GDP. So, option C is the answer for this question. So, thank you for watching my video. Please subscribe my channel Easy Life Science, like my video, share it to your friends, and don't forget to press the bell icon so you will get the notification of my newly uploaded video. Thank you once again.